I'd like very much to thank Charles Marshall, who's joined us today from the Dementia Research Centre. Charles has actually spent some time already in and out of the Dementia Research Centre, but I'm really, really pleased to say he's now about to start a project which will keep him with us for a few years, working with Professor Jason Warren, who spoke to us at the last meeting. So Charles is going to talk to us about emotion recognition in primary progressive aphasia, and I'm sure we'll be here on other occasions as we move forward into next year's agenda. So Charles, thank you so much for joining us.
Thanks very much. So I'm actually going to talk a bit more generally about the idea of doing physiological recording in frontotemporal dementia. And my research is looking at all types of frontotemporal dementia, so both the primary progressive aphasias, which come under frontotemporal dementia, so that's progressive nonfluent aphasia and semantic dementia, and behavioural variant frontotemporal dementia, which is slightly different to PPA. And I'm slightly neglecting LPA, the logopenic variant of primary aphasia today, so I'm sorry if that is neglecting anybody's interest. So I know some of you are fairly new here, and I don't want to assume too much knowledge, but I know that some of you have quite a lot of expertise. So I'm just going to talk very briefly about frontotemporal dementia as a quick reminder. So there are three main syndromes in frontotemporal dementia. There's the behavioural variant, which I've just mentioned, and that tends to be led by a change in behaviour. So people who might become disinhibited or start saying some rude things, calling people fat, or have a change in eating preference, start eating lots of sweet foods, those sorts of things as the initial presentation. And that tends to be associated mainly with loss of brain in the frontal lobes, at the very front of the brain. So if you look at picture A there, that's a horizontal slice through somebody's brain. So their eyes would be pointing up to the top and the back of their head is at the bottom. And I don't know if you can see that the front of the brain is not nearly as full as the back of the brain. You can see that the front has shrunk quite a lot. And the front of the brain is the bit that controls all those kind of behaviours that normally stop you from saying inappropriate things or being disinhibited, that kind of thing. Then the semantic variant of frontotemporal dementia is predominantly related to losing the meanings associated with words. So you may still be able to say the words, it's just that you don't necessarily recognise what they mean anymore. And a lot of that information about the meaning of words, and indeed the meaning of objects, which is eventually affected as well, is stored in the temporal lobe, and especially on the left side, because the left side of the brain controls the right side of the body. And if you're right-handed, the left side of the brain therefore controls all the important language things as well as your right hand. So a lot of we call that the dominant hemisphere, so it tends to be on the dominant left side. And confusingly, whenever we look at MRI scans, they're back to front. So it's always as though you're looking at the person. So on this scan here, this is the left side, and this is the right side. So you can see there's some asymmetry. I don't know if you can see that this here is much thinner, these bits of brain, than the right side is. So there's some shrinking really selectively affecting that left temporal lobe there, where all this information about what words mean is stored. So that's the semantic variant. And then there's progressive non-fluent aphasia. And in progressive non-fluent aphasia, the meanings of words are relatively preserved, but the thing that's most affected is the ability to actually produce the words. So it's more the movements required for speech that are affected first, as opposed to the meanings. And so these movement areas are a bit higher up. So again, because it's related to language, it's mainly affecting the left side of the brain, because that controls the language in most people, especially those who are right-handed. And so I don't know if you can see here, this left side has thinned a bit, so that the black is the fluid around the brain. There's much more fluid around the brain here because it's shrunk, but this is a bit higher up. So the semantic dementia, it's down here that's being affected, and this is the movement areas slightly higher up that are being affected, and that's why the movements required to produce speech are affected first, but the lower down areas that control the meaning of words are a bit spared. And then sometimes on this scan, it looks a bit like B, except it's the other way around. So in this case, it's the right temporal lobe which is thinned compared to the left. And because the language information is stored on the left, although it's the same bit of the brain being affected on the other side, language isn't so affected. And this also causes a slightly different version of behavioural variant frontotemporal dementia. And often people have problems when they have this right temporal lobe variant with recognising faces and change in eating habits, getting into sweet foods and things like that. A good question. So even in people who are frankly left-handed, almost all of them still have language on the left side of the brain. So if you're ambidextrous, it's very unlikely that you would have language on the right side. If you are left-handed, there's still a 90% chance that your language will be on the left side. So it's very, very rare to have language on the right side of the brain. So if something less than 10% of people are left-handed and something less than 10% of them have language on the right side, so it's probably less than 1% of people that have language on the right side of the brain. But you could, of course, if you had your language on the right side of the brain, you could have thinning of the right temporal lobe, and that would give you semantic dementia. And we're getting better and better at picking up what kind of proteins are abnormal when people get frontotemporal dementia. So the common proteins that are somehow abnormal are tau and TDP43. And something about the protein causes it to go wrong, to start folding up wrong, and to start accumulating. And when the protein accumulates, it damages nerve cells and causes these problems. And then sometimes we know that the reason the protein is abnormal is because one of the genes that produces the protein has a mutation, a genetic mutation. So a minority of people with frontotemporal dementia have a genetic problem that causes these proteins to go wrong. But in people who don't have the genetic problem, we don't fully understand why they start going wrong, what it is that makes these proteins start folding abnormally and start accumulating. So in general, I'm just going to talk about the kinds of research that you can do in dementia and in frontotemporal dementia in particular. So the first thing you can think about is how do these problems with genes and proteins cause damage to the brain cells? We don't fully understand that. We know the protein's abnormal, it's accumulating, but why does that start happening, and why does that damage brain cells? And so to do that, we test people with frontotemporal dementia for mutations in genes, and we can then take those genes and those proteins into the lab and do tests on them to work out how the gene causes the abnormal protein and how the abnormal protein damages cells. And so some people are looking at microscopic proteins in test tubes and doing things to them to see if they can get them to fold in different ways and to see how the cascade of abnormally folding protein can start. And then you can grow nerve cells in the lab in a dish, and you can see how problems with proteins cause the cells to go wrong when they're growing in a dish. And then you can try and develop animals like mice that have the problems with the proteins and see how that affects their brain. And then the next obvious question is, well, once we get a better idea of these problems with genes and problems with proteins that cause dementia, how can we alter them? Because that could be a way of finding a treatment. What can we do to stop the genetic problem that causes an abnormal protein? What can we do to stop the protein misfolding and accumulating? And so often what happens is that you get thousands of compounds and you screen them to see whether they change the protein. So you have an array of hundreds or thousands of compounds, and you add the protein to them all and you see if any of them have an effect on the protein. Then you pick out the ones that do, and then you try them in these nerve cells that are growing in a dish and see if they have an effect. And then you try them in the mice, and after 5, 10, 15 years of going through this process, you might have something that you think could be effective that you can then start working out whether it's safe in people and thinking about doing trials in people. So that's the kind of the way people are thinking about developing medications that may have an effect on the disease. And then there's the question of how can we get better at diagnosing and monitoring frontotemporal dementia? So how can we pick it up earlier? How can we differentiate it better from other causes of dementia? And then how can we tell how each individual person's frontotemporal dementia is going to develop? And so we might, for example, look at blood tests to say, well, people with frontotemporal dementia, do they have different levels of certain things in their blood that could allow us to pick the disease up just by doing a blood test? Or do the levels of certain things in their blood predict that some people are going to have a different kind of frontotemporal dementia, or frontotemporal dementia that gets worse much slower? And in the same way, we might be able to find certain things on scans that can give us those kinds of clues, or even psychological tests. If we could find very good psychological tests where if you don't do so well on them, that's a very, very early sign, that means we can pick up the disease really early, then that's also very useful to us, especially when we do find something that we think might be a treatment, because then we can start it very, very early, because the earlier you can start a treatment that's being developed, the more likely it is to be beneficial, because once you've got lots of symptoms, you've lost quite a lot of brain cells that don't grow back, so you need to stop the damage to the brain cells before that happens. And then there's another kind of research, which is more the kind of research that I'm focusing on with Professor Warren, which is which bits of the brain are affected in frontotemporal dementia, and how do these bits of the brain being affected cause the symptoms that we see. And we call this the systems neuroscience of dementia. So what we're trying to understand is, we know, for example, that people with frontotemporal dementia sometimes, as I've said already, have a preference for sweet foods that develops. But why is that? Which bits of the brain that control eating behaviour and taste preference are affected? And what can that teach us about what's happening in frontotemporal dementia? So we know that those proteins that I was talking about that go wrong are found all over the brain. But why, if they're found all over the brain when they go wrong, do very specific brain areas get affected and not others? And that's the real mystery that we're trying to work out. So part of that is getting a much better idea of which brain networks are affected. So I'm going to talk a bit more about this kind of systems neuroscience, this approach to really pinning down which areas of the brain are affected and in what order, and how that causes symptoms and changes in perception and behaviour. So the kinds of tests that we normally do in this systems neuroscience research, some of you will have quite a lot of experience of. So questionnaires are quite a big thing, actually, because looking at what symptoms people have is very useful. So you might say, have you noticed a change in your food preference? Have you noticed a change in the way you appreciate music? Are you much more into music, or does it just not do anything for you anymore? And that's interesting to us to know. And then there's more kind of methodical behavioural testing. So you might test somebody's ability to name objects or recognise facial expressions or something like that, kind of psychological testing. And then once you've got the results from the questionnaires and the behavioural testing, you can then go on to look at brain scans, and you can get a computer to do a complex analysis and say, well, if we compare all the scans of people that have started liking sweet foods more with all the people that are just the same in their taste, which bits of the brain are smaller or bigger in the people that have had a change in food preference? And then that tells us something about which areas of the brain might be responsible for that. And then there are functional scans. This is where you're doing some of the behavioural testing lying in the scanner. And the MRI scanner can light up bits of the brain which are using more oxygen and therefore having more activity. And so this is a better way of pinning down exactly which bits of the brain are active when you're doing something. So you could show that people are recognising faces, and if you have more shrinkage of certain bits of the brain, you're less good at recognising faces, but then you can then go on and show that the bits that are shrunk are the same bits that light up when you're doing that task, and therefore you're getting really strong evidence that these bits of the brain are responsible for that particular task. But I'm thinking about something different to these types of tests that we've been doing. I'll just tell you about this. This is an example of recognising emotions. This is from the Dementia Research Centre. And this was a test where people were asked to identify which emotion was expressed by a piece of music or by a facial expression. And we then got the scores together for how many people got right, and then got the computer to look through their brain scans and say, well, if you did worse at recognising emotions, these are the bits of the brain that were shrunk more. So it's quite a blurry picture, but the bits in red are the bits that are more shrunk if you're not so good at recognising emotion. And these 6 are in music and these 2 are in faces. So this is the kind of thing I'm talking about that gives us a clue to which brain areas are affected and which are causing the problems. So I'm interested in measuring other things at the same time. So these physiological recordings, so monitoring changes in the body, and that's things like measuring heart rate, measuring the change in conductance of your skin. So if you're aroused by something, if you're enjoying something, or if you're stressed and nervous about something, you tend to get change in your skin, which is what people are measuring with lie detectors. That kind of stress that you get, that they can measure by measuring your skin. And also pupil size, so if something makes you happy or alert, or if something makes you anxious, then your pupils dilate. And it's, you know, what poker players look for when they know that somebody's scared because they think they're about to lose the hand and they spot the pupils dilating. It's that kind of thing that I'm looking at measuring. And also muscle activity. So this is what I'm going to talk quite a lot about, the way in which somebody can actually reflect in their own muscle activity what they're experiencing and whether that would be useful for us to measure. And the reason to measure these is that you get a nice objective measurement. So, for example, if you are playing somebody music and looking to see how much they enjoy it, you can ask them on a scale of 1 to 10 how much you could enjoy that. But it's very kind of arbitrary rating on a scale like that. And some of the participants in our experiment have difficulty actually pinning things down on that kind of scale of measurement. Or, you know, sometimes they don't completely understand the question. And so it would be nice if instead of getting them to rate on a scale of 1 to 10, you could measure their pupils and say, well, I can just tell by how big your pupil got, how much you liked that music. It would be a nice objective measurement, and it would mean people wouldn't have to spend so much time, you know, rating scales of 1 to 10 things, which also gets very boring. People get very fed up with research when you get lots and lots of boxes to tick and things. And you can measure the change very rapidly. I mean, you can measure these changes over milliseconds, whereas that functional imaging that I was talking about, looking at which areas light up, you can only measure that over a few seconds, so you're not getting a very precise snapshot. And it may shed new light, measuring these things, on how problems with perception arise in frontotemporal dementia. So it may be that the reason that you don't always understand emotions and perceive emotions so well is because you're not actually taking on those changes in your own body. You're not changing your heart rate and your pupils and all those things, and therefore that makes it more difficult to work out what emotions are. And it may show that this thing that I'm about to talk about, embodied simulation, is important for making these sort of complex emotional judgments, like what the composer meant when he wrote a piece of music, what was the emotion that he was trying to get across. It's a very complex thing, that, and a very complex process that you know, humans have evolved recently that allows them to understand subtle emotions in each other. And there's a theory in neuroscience that this relates to embodied simulation. And I'm sorry this is a bit long, I'm just going to read through it slowly, but I think it's brilliant because it shows that the philosopher Nietzsche was about 120 years ahead of neuroscience in thinking in this way. So he says that in order to understand our neighbour, that is, in order to reproduce his sentiments in ourselves, we often no doubt plumb the cause of his feelings, as for example by asking ourselves, why is he sad, in order that we may become sad ourselves for the same reason. But we much more frequently neglect to act thus, and we produce these feelings in ourselves in accordance with the effects which they exhibit in the person we're studying, by imitating in our own body the expression of his eyes, his voice, his gait, his attitude, or we may at least endeavour to mimic the action of his muscles and nervous system. A like feeling will then spring up in us as the result of an old association of movements and sentiments which has been trained to run backwards and forwards. So what Nietzsche's saying is that instead of saying, well, if I was already running late for work and then I got splashed by a car and then I got a parking ticket, how would I feel? Sometimes we do that. We say, how would somebody feel in this situation? But much more often, we pick up on subtle changes in facial expression, body language, and we replicate them in ourselves. And then we get a direct emotional contagion. So without having to do that slow thing of thinking, how would I feel in that situation, we're actually taking on the other person's emotions, and that's allowing us to understand them. So for example, if we see somebody's face make an expression, our face mimics it. And sometimes this is very subtle. So sometimes you're talking to somebody and they smile and you smile, and that might partly be a social thing, but we're doing this in a way that you can't always detect by looking at somebody's face. It's a subtle change in the muscles that you can't actually see. And it's involuntary. So people have done tests where they've measured the activity in people's facial muscles and they told them, whatever you do, don't move your face, don't mimic the expression, and watch videos of somebody else having a facial expression. They can't stop it. You can still tell from recording in the muscles that they are involuntarily mimicking those expressions. And so the idea is that you're then looking at your own body to work out how you're feeling. So you're saying, how would I be feeling if I move the muscles in my face in this way? And so the questions we're asking are, when people with frontotemporal dementia have difficulty recognizing emotions, do they still get these changes in their facial muscles? And also in other things like their heart rate, because you tend to move your heart rate up or down in response to other people's emotions as well. Or do they do these things, replicate the emotion in their muscles and their heart rate, but they become less sensitive to changes in their own body, and therefore they're not quite picking up on those signals from their own body? We're not quite sure which of those is the case, so this is what I'm looking at. So my current experiment I'm going to tell you a bit about. The first part is heart rate counting. So this is a slightly funny task where I get people to sit quietly and just see if they can feel their own heart beating. Not by taking their pulse, but just sit. And that feeling you get when you've run up the stairs and you can feel your heart pounding in your chest, they have to try and tune into a much softer version of that and see if they can feel it. And at the same time that they're trying to count their heartbeats, we measure how many heartbeats they're actually having, and then work out the percentage of the heartbeats that they were able to feel. And this provides a measure of how sensitive somebody is to what's going on in their own body. So then we can look and say, well, if people are more sensitive to changes in their own body, are they better at recognizing other people's emotions? And certainly in young normal people, this is the case. So the better you are at feeling what's going on in your own body, the better you are at recognizing emotions. I think it'd be really nice to look at that in people with frontotemporal dementia as well and see if the same thing holds true. And then the second part of the experiment is where people watch videos of facial expressions. And then after each video is finished, they have to say what they thought the expression was: anger, fear, happiness, or surprise. And while they're doing this, I'm measuring the electrical activity in the muscles of their face. And in particular, I'm measuring the corrugator muscle, which knits the brow when you're frowning, and the zygomatic muscle, which raises the corners of the mouth when you're smiling. And then looking back to see how much the activity was changing when they were seeing the different facial expressions. So this is my friend who was helping with the testing out the experiment. This is what people look like when they're doing the experiment. It's wires stuck onto the muscles of the face with a bit of tape over, and then I sit them in front of a laptop and they watch the videos. And these are just some examples of the videos, if I can get them to play. There we go. So this is an example of a video for anger. This is an example of fear. You may notice that you're mimicking the emotions a bit as you watch them now. And this is happiness. It's quite hard not to smile at that one, isn't it? And surprise. So those are just some examples. And people watch about 40 or 50 of these with all the different emotions. And so this example, this is when I was testing it out in younger people. I'm sorry, it's not projecting very well, but so this is in young normal people, and this is just looking at anger. And these lines here are the muscles that knit the brow, the corrugator muscles. And this one here is the muscle that makes you smile. And you can see that during the video, this is averaged over lots of videos and lots of people, but you can see that they're really knitting their brow. The activity in those frowning muscles is going up as they're watching. And the activity in the smiling muscles isn't just doing nothing, they're actually stopping their smiling muscles from working, they're depressing activity in those muscles. So you can really tell. And what was interesting about this is that I got the people to hit the space bar when they knew what they were looking at. And the time they pressed the space bar is the end here. So actually you could tell from looking at their muscles what they were seeing a second and a half before they knew what they were looking at. So actually you're doing this very early on when you're looking at these emotions. And the fact that you're doing it so early helps to support this idea that you're actually doing that and then examining your own muscles to work out what it is that you're seeing. And this is just some more graphs. So this is these ones going down, the green and the purple, are happiness and surprise, and the ones going up are anger and fear. And this is the activity in the frowning muscles. So you can see when people are looking at anger and fear, the activity is going up above zero in their frowning muscles. And when they're looking at happiness and surprise, they're doing the opposite. They're suppressing the activity in those frowning muscles. And again, the time when they knew what the emotion was is at the end of the slide here. This is all before they knew what they were looking at. And then this is the opposite. So this is the activity in the smiling muscles, and you can see it's going down for anger and fear and going up for happiness and surprise before they know what they're looking at. So this was just to start the experiment with healthy controls and make sure that it worked. So now I'm going on to do this experiment in people with FTD and age-matched controls. And I'm also going to do this heart rate counting task and look to see, so it may be that people with FTD are a bit less sensitive to their own heart rate, which would be interesting in itself. And it may be that within FTD, the more sensitive you are to your heart rate, the better you are at recognising emotions, and that it may be this system which is being affected. And it may be that people with different types of FTD perform differently at this. So people with progressive non-fluent aphasia that have those movement areas affected, the areas that control movement of speech and in the face, may not be able to imitate so well with the muscles because it's those bits of the brain that are affected. Whereas people with the behavioural variant of FTD, the motor areas are working okay, so they might imitate, but then the next bit that you have to do, which is then working out from imitating what to label that emotion, they may be less good at. And then I'd like to try and do this with some functional imaging, so do the same test but do it in the functional MRI scanner, so I can look and see which bits of the brain are active when you're imitating and which bits are less active when you're not imitating the expressions so much. And then I could go on to add other types of emotion recognition, so get people to identify emotions with sounds and music and things like that at the same time, and see whether it's just facial expressions that make you activate your muscles in your face this way, whether it's also music or other sounds. And the idea is to then look and see which brain networks are affected, and then to go back and think, well, now that we've got a better idea for which brain networks are affected, why are they affected? Why are these protein problems causing this? And what I've started thinking about is maybe the reason that these particular networks get affected first in FTD is because they're more recent networks. They're the most complex things that we've evolved to do, to identify subtle emotions, to speak, all these things that have only happened in the last, few 100,000 years to the human race, whereas the much older things that have been around in other animals for much longer maybe are a bit more robust, and maybe that's why these networks are being affected early. And the protein problems seem to pick them off first. So I just want to thank, I'm funded by a philanthropic grant from the Leonard Wolfson Foundation. He had dementia and bequeathed a large amount of money that UCL put in a bid for and used to set up their Leonard Wolfson Experimental Neurology Centre and the PhD programme. So I'm very grateful to that opportunity to be able to do this research. And then the people at the Dementia Research Centre who have been very, very supportive and friendly. It's a really wonderful place to be. Professor Warren, Professor Fox, Dr. Rora, Camilla Clark, who I think lots of you will have met, Chris Hardy, who I think some of you will have met as well, and Katrina, and most of all, the brilliant participants, the people that have come and given their time and put up with me sticking things on their face and so on. It's really amazing to have people who are so prepared to do all these things for us. And just to say that if you're keen on research, if you're curious about it at all, you can talk to me afterwards, you can email me. And there's no obligation at all. If you want to find out what it's about and say, I don't think I fancy that, that's fine. If you want to find out what it's about and say, well, I'm not sure I want to do that bit, but I'm happy to help with another bit, that's fine as well. And there are quite a few different strands going on at the Dementia Research Centre. So often what we do is try and bring people in for 2 days, either consecutive days or separate days, whenever suits you, and timetable in different things, scans, a medical assessment, blood test, other testing, so that all the people doing their different research strands can get the information that they need. So come and find me afterwards or send me an email if that's something you think you might like to be involved in. Okay. Any questions? So have I got any preliminary ideas as to what sort of results I'm getting? The answer is no. The reason being that with looking at this kind of muscle activity, you can't really tell from any individual person what the effect is. You have to look across quite a lot of people and get the computer to do averages and things to see whether it's working, because the muscle activity is all over the place. So you have to look over lots of people and see if on average it's changing. And I've been encouraged that it's bad science to keep looking at your results and peaking, because say you looked and you had a nice effect, you might then say, okay, well, I won't see any more people then, because if I collect more people it might disappear. So it's better to collect lots of information and then look at it and try and be as unbiased about it as possible. So I'm resisting the urge to peek at my data right now. But I've seen about 14 or 15 people so far, about half FTD and half controls. So when I get up to around 20, I will be unable to resist the temptation to have a peek at my data then, I think. Do any volunteers? The answer is yes, we do. We need people who are age-matched controls, so people between about 50 and 80, usually we say. And we have some controls who just come in for one visit to do a couple of things, and we have some people who are willing to give up lots of their time, who come back regularly. We have some people who say, I'll come and do some of the testing, but I don't really want to have a brain scan because I don't really like it. So anybody who's willing to contribute in any way, please let us know. We're always keen to have people who are willing to come. And some of it's quite interesting and quite fun as well. Some people quite like it, get a bit of insight into what's going on. We tend to bring in people with FTD from our clinic, Cognitive Disorders Clinic at the National Hospital for Neurology, but we're always willing to have people from elsewhere. And there are two ways of doing that. Either somebody's neurologist can send them to us, or you can contact us directly. But I think in either case, just to make sure that we're kind of on the same page, we'd normally want somebody who we haven't seen in our clinic to come to a research clinic and just be assessed before enrolling, just to make sure that, you know, our idea of what constitutes behavioural variant or progression of aphasia is the same as someone else's, so that we know that we're being consistent. It's a good question. So none of the dementias yet have a treatment that actually changes what's going on under the surface. So this process where abnormal proteins accumulate in nerve cells and damage them, we don't have anything yet that can stop that. Some dementias have symptomatic treatments. So, for example, in Alzheimer's disease, whereas it's a different set of nerve cells that are affected. So these bits that control emotional processing and language and behaviour aren't so affected. And the nerve cells that are affected are nerve cells that produce a chemical called acetylcholine. So the treatments that are available for Alzheimer's are simply medications that boost the level of that chemical because it's slightly deficient. And so that just makes symptoms slightly better, but it's not doing anything to reverse the underlying process. And so we're trying to come up with these sorts of treatments, and we're trying to look for chemicals that stop proteins folding abnormally. But part of the problem is when we've tried these things, they haven't worked. And we're starting to think, well, maybe that's because the people that we're trying them in have had so much protein build-up. You've had a lot of abnormal protein build-up by the time you start to get symptoms. And maybe to really stop things in their tracks, you need to start much earlier before somebody develops symptoms. But that's very tricky, because how do you identify people before they've noticed there's anything wrong with them? This is our real problem at the moment, and this is what we're working on. So I was talking earlier about a new study that launched this week, thinking mainly about Alzheimer's, but also because it's common, but also about other kinds of dementia, which we're running at the Dementia Research Centre, which is the 1946 study. So this is a group of people born in the same week in March in 1946, who've been followed their whole lives, collecting all sorts of medical data and educational data and all this kind of thing. And so they're now 69, and we're starting to bring them into the Dementia Research Centre and do scans and blood tests. And then some of them, unfortunately, over the next 10 years or so will develop Alzheimer's. And then we can look back and say, well, was there anything that could have predicted who was going to develop it and who wasn't? And I mean, the really wonderful thing is if we could say, well, there was this blood test that we did, and when people had a positive blood test, they went on to develop Alzheimer's, and something like that, we could then start using to screen people in the population. We could say, like with having a mammogram or a test for colon cancer that's sent out now, you could have a blood test that could determine your risk of Alzheimer's, and if it's positive, then you start a treatment. That would be the ideal, but we're quite a long way from that. And the difficult thing is that if you say to somebody, well, we've done a test that shows that you're not definitely, but there's a chance that you're going to get Alzheimer's, and there is a treatment, but the treatment has terrible side effects, do you want it? It's all very difficult. So we're kind of working on these things, but these are the kind of complexities that mean it takes a very long time. Yeah, so exactly. So in both in Alzheimer's and in frontotemporal dementia, you can see changes in the brain. And some types of frontotemporal dementia that are related to genetic mutation in the C9orf. You can see changes in the brain going back 30 years. But certainly in Alzheimer's and other kinds of frontotemporal dementia, it's more than 10 years that you can see subtle changes and the beginnings of the accumulation of the abnormal proteins. We need to start trying to nip things in the bud. That's a good question. I think there's a tendency for people with any kind of dementia to be told they have Alzheimer's because it's so common and because it can present with different things, and it's easy coming from the kind of ivory tower of the Dementia Research Centre to say, oh well, you should have been diagnosed with frontotemporal dementia all along, but we have quite a lot of expertise and we're quite protected in our ivory tower, and that we have a lot of time to spend with people to really get a feel for the exact problems that they've got. And somewhere else where people are much busier and have less privileged in terms of the time they've got and the expertise they've got, there is a tendency to just say that you've got a dementia or you've got Alzheimer's. And I think that's going to have to change because the way that we think about dementia is changing, because we're trying to develop treatments, because we're trying to study these diseases in much more detail. We're going to need to diagnose people correctly and early. And so I think that we need to train people up better for that, and we also need to have more money in dementia services so that there are more doctors, more nurses, more psychologists, more time that we can spend with people to get the answers right. And then on the other side, there are some of those types of research that I was talking about where we look at ways of getting better at our diagnosis. So if you could come to the doctor with some complaints about memory or word finding or something, and they could just do a blood test and say, well, the blood test shows that you haven't got Alzheimer's, you've got frontotemporal dementia, that would obviously make life much easier as well. So all those things are important. And also because dementia is often quite subtle to start with, and it takes time to develop. So, we at the Dementia Research Centre often see people who've had symptoms for a few years, and then they come to see us and we say, well, it's so obvious. Well, sure it is now, but it wasn't. It was much subtler when you were first seen by somebody elsewhere. So. It's not a hard and fast rule. It's really just reflecting the rough age spread of our patients, because we want the average age in our controls to be about the same as the average age in our patients. And unfortunately, it's a fact that people who are older are less likely to make their way to our services, because if you're older, you're more likely to see someone in old age psychiatry who says you've got Alzheimer's, and whereas if you're younger, people often normally worry a bit more about the diagnosis. Unfortunately, it's just a sad fact, and we tend to have an unrepresentatively young population at the Dementia Research Centre. I think it's okay. I don't think it's going to have an impact on the way that your brain is functioning, except that if you're a bit sleepy, you're a bit less switched on. But if you don't take it, you'll be back to normal. I don't think it'll affect you adversely. Sometimes the PPA is related to Alzheimer's, and usually that's the logopenic variant, which I've neglected slightly. So the logopenic variant of primary progressive aphasia is often an unusual way that Alzheimer's develops. Yeah. But most PPA comes under the frontotemporal dementia spectrum. And we're sometimes caught out. We sometimes have people who are good enough to donate their brains for us to look at after they've passed on. And sometimes we look back and we say, well, we thought they had frontotemporal dementia, but actually it was Alzheimer's all along. But in general, PPA is mostly caused by frontotemporal dementia. And part of the problem is that the medication that we give for Alzheimer's is only improving symptoms, so it's not actually changing the course of your disease. And often when we give it to people with frontotemporal dementia, it makes them worse. So that's why there's a bit of caution, because when those nerves that are making acetylcholine are actually fairly normal, and then you boost the levels of acetylcholine, it can have quite marked effects on people, and it can be quite unpleasant. So that's why we're a bit wary of giving that out. I think it would be different if we had something for Alzheimer's that was actually changing the course of the disease. Then we would probably find ourselves being much more anxious about it. At the moment, whereas if you're not having the Alzheimer's treatments, you're not missing out on that much, because they only make a very small difference symptomatically. If you have the logopenic variant due to underlying Alzheimer's, then the medications might help your memory a bit, but they won't help the aphasia. That's the other thing to say. They don't really. So I'm a neurologist. I have nearly finished my clinical training, so I've got 6 months of working in the hospital left till I'm a consultant, but I've stepped away from that for 4 years to do a PhD. And I'm a year in, so I'll be doing research at the Dementia Research Centre for my PhD until summer 2018, and then back to the hospital for a short while before I start looking for a consultant job.
If I could follow up on both of these questions about
Please.
diagnosis, because I don't know, I've been coming on and off, it must be on for a couple of years here, and one of the things that I have learned from people here is so many people are saying the diagnosis is wrong. We had the lady, the clinical psychologist, who came along and stood up there for a year and said, lots of doctors don't know anything about FTD. You said it. There was a gentleman who came, I think it was the first time we came there, and his wife had been diagnosed 2 years previously and with Alzheimer's, and he was fighting. So this is going on all the time, and obviously, I'm convinced that my sister was wrongly diagnosed. When she went along to the memory clinic, they tested her for 90 minutes or whatever it was, and because what the people here had told us, her doctor kept on sort of throwing in FTD and whatever, and so that it wasn't as if it wasn't even flagged up for them. And they came back with a specialist social worker, not even a doctor, came and told her she's got Alzheimer's.
Wow.
And so somewhere along the line, the medical profession's fallen down here.
I agree. I think that there are lots of reasons for these things. So one is that the disease evolves, so sometimes it becomes clear later on what's happening when it wasn't completely clear at the start. Another is that because all of medicine is getting so complicated, it's very difficult for people to have specialist expertise in lots of areas. So whereas some years ago a neurologist even within neurology could know most things about neurology, now we know so much more about the diseases that even within a specialty like neurology, people have to have their own expertise. So there are people who are experts on muscle or spinal cord or movement disorders, or people who are experts on dementia. And so, but even short of being a neurologist, for a GP to have the kind of expertise to understand the subtleties of the difference between different kinds of dementia is very difficult. And a lot of the time people with cognitive problems are being seen by psychiatrists who have, amazing expertise at looking after psychiatric conditions, but the memory bit is sort of somewhere in a grey area between psychiatry and neurology, and especially if you have the problems with aphasia, that's much more neurological. So if you're seeing a psychiatrist, because that's historically the way that people have gone to be assessed with dementia, it's for them to work out the subtleties of your language problem is very difficult because it's not what they've been trained for. And so we probably need more neurologists to be out in the world seeing people with the benefit of their specialist training and getting this right, but we don't have enough neurologists in the country. So we have something like less than half the neurologists per head of population of most European countries. We just don't have enough. The Dementia Research Centre, it's all we do, and as I was saying before, we have this amazing luxury of time to spend with people, time to think about things and work it out, that in other places they just don't have. They're much busier and much more swamped. And even then we get it wrong, and we see somebody and we have an idea, and then we see them again and we get a completely different feel for it. It's complicated and it's subtle, and it's not always easy to get right. So identifying that there is a problem is definitely something that the memory clinic would do, but working out the exact nature of the problem, which bits of the brain are affected, what the underlying pathology is, it's complicated and it's difficult. I think part of the problem is that neurologists in the UK have historically tried to maintain a certain kind of practice where they did very specialised things, and to justify having more neurologists, you need people to be prepared to spend more time in A&E, more time in the stroke ward, more time doing the kind of bread and butter things. And the problem is neurology attracts people that like seeing weird and wonderful things, and they want to try and keep themselves seeing weird and wonderful things. And unless neurologists are prepared to, really engage with the rest of the medical world at a more fundamental level, it's hard to justify there being more of them. So it has to happen from both sides. Neurologists need to embrace doing more, and we need more neurologists to come and join in both sides.